All right. Good afternoon, physiology. This is our last Zoom meeting. I'm excited about it. All right, so next time we meet, we're going to be meeting in person. And today's lab worksheet, we're going to do part of it as a class, and then you're going to do parts of it on your own. And it is due on Canvas before midnight on Sunday. So since we're going to be in person next week, we're going to start next week with a discussion of the things that are going to be different when we're in person compared to virtual. Things like the lab notebook, which I haven't had you guys keep yet. We'll start keeping that next week, and I'll talk about how we're going to do that and how we're going to grade that. There are going to be some changes in the original grading plan because we did miss so many in-person labs. And we really need to do a lot of skill learning in the lab where we're actually practicing with skills. So there are going to be some changes there, but I will explain all of that next week when we're in person. Now, some of you may not have been to campus before, or at least not during the pandemic. So I thought I would start with this. What do you have to know to come to campus? So there is an app. It's called My BCCCD. And you'll need to upload that onto your phone if you have a smartphone. If you don't have a smartphone, you can do that part once you get to campus. I'll explain that in just a second. So you download the app, you upload your COVID vaccine information. The college is not requiring boosters, although I strongly recommend them. But you will not be allowed to come to class if you are not vaccinated. So you have to be vaccinated to come in person. When you come to campus, every day, you're going to open that My VCCD app. It's going to ask you some questions. You're going to answer them, and it'll give you a notification saying whether or not you're allowed to come to campus. Then you're going to go to one of the check-in locations. A lot of them are in the parking lot, and you show your notification to the person there. Now, if you don't have a smartphone or your smartphone can't handle the app for whatever reason, they'll have a tablet there that you can use to fill the same information out. They will then give you a wristband that you must wear while you are on campus. And the color changes daily, so you get a new one every day. We are also supposed to be scanning QR codes to keep the college up to date on what buildings and classes we've visited. So if you go into a building and you scan the QR code and somebody in that building reports that they have COVID, you'll get a notification. In addition, we are requiring masks. I know the state is lowering the rules, but personally, I think that's a bad idea, and the college is still requiring them. They have to go over your nose. If your mask is down here, it's not doing anybody any good. If you have problems getting your mask to fit comfortably or fit well, let me know. I've got some tips that might be able to help. Same thing if you have a lot of fogging up the glasses. I've got a few tips that help. If you like to use those neck gaiters that look almost like a bandana around your neck, those are not masks. Those don't really help very much. If you don't have masks, I can get you some surgical masks, just have to ask. What I'm doing right now is I'm double masking. So I'm wearing a surgical mask and then a cloth mask over it. The N95s are currently, you can pick those up. You can pick up a couple for free at most drugstores in the area. I find that they tend to get caught in my hair. So that's why I'm going with the double masking instead. Your mileage may vary depending on your hair texture and the amount of hair you have. Any questions about how to come to campus? Looks like we got it. Good. All right. Next week, we are going to be learning how to do dilutions. And in order to do dilutions, we have to be able to pipette. We're going to walk through this in lab next week. But I wanted to spend a little bit of time talking about pipetting and showing you a quick video to kind of give you the advance notice of what we're going to be needing to be able to do next week. So what I'm going to do because the way my computer is set up, I cannot play audio for you. So I'm going to put the URL in the chat. It's a 3.5-minute video, so I'm going to give you 5 minutes to go watch that video, and then we'll all come back and continue on with the lesson. So 5 minutes. Okay, so hopefully that was useful to you guys. I want to make sure I emphasize a couple of things real quick. You must use a tip. So the pipette itself is this mechanical structure here, but on the end you can see the plastic tip. You always have to use a tip. And those tips are single use and disposable. When we get to the lab, there will be a container on your bench that you use to dispose of those lab tips. Never adjust a pipette outside of the range. What do I mean by this? So the pipettes are called by the maximum volume that you can use that pipette for. So a P20 can be used for 20 microliters. A P200 can be used for 200 microliters. A P1000 for 1,000. So if you need to weigh out or measure out 201 microliters, you can't just set the pipette for 2001. You can turn the knob and force it, but that will break the mechanism. So you want to go up and use the next larger size. We are going to be practicing pipetting in the lab next week. So we'll go over some of this stuff again, and we'll be able to get our hands active and doing stuff. It's going to be exciting. But our last Zoom class is going to be on sensory physiology. So when we think about sensory physiology, we're talking about several different things. Part of it is homeostasis. You guys remember homeostasis? Good. Okay, so if you're trying to maintain the pH of your blood at the right range, you have to know what the pH of your blood is. Do you know what the pH of your blood is right now? If you think really hard, no, you can't tell, but your body can tell. And so it's going to use a sense to do this. And it's not a sense that we are conscious of. So specialized cells, they're sometimes called receptors or sensors, detect a physiological variable like blood pH, like blood sugar, like the salinity or the amount of salt in your cerebrospinal fluid. And they trigger a response to maintain homeostasis. If your blood pH gets out of whack, one of the things that you can do if it gets too high is you start to breathe faster. That gets carbon dioxide out of your bloodstream, and the carbon dioxide in your bloodstream can screw up the pH. If the salt concentration in your cerebrospinal fluid gets high, you feel thirsty. So you drink water and you can dilute that cerebrospinal fluid that way. And the muscle reflexes are one of these sensory physiology mechanisms. If your muscle length changes suddenly, and it's not like you're stretching the muscle, but like you get punched in a muscle or you trip and kind of almost overextend your muscle, that can trigger a contraction of the muscle to help protect it from tearing too much, to reduce the amount of damage that's going to happen there. Now, when we think of senses, we generally think of these 5 senses, right? This is what we all learn when we're kids. We have 5 senses. Well, these are the special senses. And that means there is a specialized sense organ. And we are aware of these. So we're talking about vision, hearing, smell, taste, and touch. Those are not all of the senses we have. Depending on how finely you separate things, there are a lot more. So touch, for example, there are several different types of sensors that detect different types of touch. So a light touch versus pushing down on your skin, those are different sensors, even though we call them both touch. And then these homeostasis associated receptors, those are involved in what we call the general senses. There's no specialized organ. And often these are things we are not aware of. We're not consciously thinking about that. Although hunger and thirst are examples, we do get thirsty, right? But are you aware of what you're doing when you're working on your balance? Most of us aren't, unless we're doing yoga or something. How about proprioception? Do you even know what proprioception is? I bet not. Proprioception, the best way to define it is body awareness. So I'd be willing to bet that without looking, you could tell where your feet are. You know where they are. When you're walking, you don't have to consciously think about every part of the movement. Bring the leg forward, bend the knee, put the foot down. You don't have to think of that. That is all involved in proprioception. But those other things that I talked about under homeostasis, blood sugar, pH in various organs, oxygen availability, those are all things that fall under the general senses. Okay, if you need to take a screenshot, now is your chance.
Professor,
Yes.
for proprioception, is it the same sense then when you choose to be aware of it, or is it only the unaware part?
It's the same sense when you choose to be aware of it. Yeah.
Okay.
And good proprioception is what separates the professional athletes from, the rest of us.
Yeah, I was about to say that.
Yeah.
Yeah.
You're exactly right there. So there are several different ways we categorize receptors. And I'm going to start with the type of stimulus. So what triggers the sensor or the receptor to respond? So we can divide all of these receptors and sensors into 4 categories. Mechanoreceptors, think mechanical. Those respond to physical pressure. Thermoreceptors that respond to temperature. Interestingly, we have different types of sensors for hot versus cold. It's not one sensor that detects both. Photoreceptors for light, and chemoreceptors for chemicals. So, when you think about mechanoreceptors, of course you're going to think about touch. But this is also what's used for hearing, equilibrium, and proprioception. All of those are actually involve physical pressure. When you get to senses in the lecture, I'm sure you will learn about the little tiny stones in your ear that are used for balance, that help your body understand which direction gravity is pulling. And you'll see that I've got pain written here. Pain, sometimes called nociception, fancy word for it. If any of these sensors get overstimulated, the signal that you get is pain. So if you touch yourself on the arm, no big deal. If you push really hard, eventually you'll get to the point where you feel pain. The thermoreceptors, that's just temperature. Do you feel hot? Do you feel cold? Again, though, if they get overstimulated, you get pain. Too much cold or too much heat hurts. With photoreceptors, that's obviously vision related. There are multiple kinds of photoreceptors, some that work best in low light, some that are involved in color detection. And again, look right here, it says pain. If you've ever gotten a bright flash of light in your face, you're driving down the road and somebody turns the corner and comes at you and they've got their high beams on and it hurts. It's overstimulation of the photoreceptors. And the last category, the chemoreceptors, boy, I've got a lot of spelling errors in this one. Smell or olfaction and taste, but also a lot of those general senses. This is how your body knows what your blood sugar level is, what your oxygen concentration is, what the pH is. Oh, and look here, pain as well. So those are the kinds of receptors that we're talking about, the different categories based on the stimulus. I'm going to dart over to the whiteboard for a second. And I want to talk about very simplistically how senses are perceived. So you've got your receptor, got your little receptor here. Actually, that's not the receptor. Okay, so you've got your receptor. And the information from that receptor travels a pathway to the brain. So it receives a signal here, goes to the brain, and it goes to a specific location in the brain. It's not just going to go anywhere. We call the way the information goes from the receptor into the brain a pathway. That makes sense, right? And pathways seldom change. And if they do change, they change slowly. So you can certainly change these pathways by doing things like practicing. If you want to learn how to juggle, the first, probably dozen times you practice, you're dropping a lot of balls. But eventually you get the rhythm going and you can juggle successfully. That's an example of a proprioception pathway change. Now, any kind of stimulus that triggers the receptor is going to go along that same pathway. So let's think of our vision. Generally speaking, with vision, it's light hitting the receptor and that information goes to the brain. Well, have you ever rubbed your eyes really hard and seen flashes of light? Those receptors are being triggered by you rubbing your eye. And they are sending a signal to the brain, and the brain is like, this came from the eye. It must be light. And so your brain kind of makes up what it thinks might be going on. There's some really weird stuff that goes on with vision if you really get into it. We can also talk about this with another example, and that would be hot sauce. If you like hot, spicy foods, a lot of those chilies, jalapenos, and that sort of thing, there's a molecule called capsaicin. And capsaicin doesn't go to your taste receptors. Capsaicin binds to your heat sensors. So a thermosensor is actually that chemical bonds to the thermosensor. And so it kind of feels like, well, it feels hot because the heat sensor is going to send a signal to the brain saying it's hot, even though the temperature hasn't changed. It's been triggered by something that is not what it's designed to detect. We doing okay with this? Good. All right, I'm going to clear this. Because I want to introduce another way we categorize receptors, and that is by activity type. So all receptors, sensors, will be one of those types of physical senses, and at least one of the activity types. So it's kind of like saying all dogs will have one color type and one fur length type. They're two different ways of kind of organizing them. And so our activity types, they've got fancy words, tonic and phasic. Tonic receptors respond as long as there is a stimulus. So a lot of pain receptors are these way, are this type. If you think about it, you break a bone in your leg, hurts like hell. If the receptors quit sending the pain signal to your brain, you might get up and walk around and think, oh, it can't be hurt that bad. I'm just going to walk around. You don't want that to happen. So that sensor is going to constantly signal the brain, and your brain is constantly going to say, oh my God, I broke my leg and it hurts. The other kind is phasic, and those respond to changes in stimulus or stimuli. So if constant stimulus is applied, the sensor adapts and quits sending the signal to your brain. And if you want to think of an example, are you aware of your clothes right now? Do you feel the weight of your shirt on your shoulders? You might now that I've mentioned it, but we adapt to that, right? We wear our clothes all day. You might notice when you pull them on how they fit, but then you just kind of, your body gets used to feeling it. And that is a phasic response.
Hey, Professor, is that the same thing as olfactory nerves, where they get used to the smell of your environment?
Yes. Yes. Olfactory is a great example of it. If you've ever walked near somebody or stood near somebody who had just insane amounts of perfume or cologne on, it's probably because they kept reapplying it when they quit smelling it. So yeah, that's another really good example. Okay, how are we doing with all of this? Seem to be doing okay. All right. I'm going to clear this. So I hope you guys have either a digital or a paper copy of the lab activity worksheet. You can either record your answers digitally and upload the worksheet, or you can record on a printout, just write it out and take a photo to upload that. If you're just sending me the file, please put your last name in the description and what you name the file so that I know whose file I'm getting. Now, not everybody is going to be able to complete all of the exercises as intended. This is fine. This is okay. No big deal. If you're having particular problems with one of the activities, just let me know. We can see if there's another way for you to learn the information there. So we're going to start with vision. And obviously, if you don't have great vision, you might not be able to complete these exercises. If you have problems with your arm mobility, there's one of the last ones that we're going to do as a group. You might have problems with that. If you get weird results, use Occam's razor. The most likely thing that's going wrong is how you're doing the experiment. Not that there's something wrong with you and you're dying. So keep that in mind as we go through this. So the first thing we're going to talk about, and the first thing we're going to do, I'm going to talk about each of the activities, even if we're not doing them as a class. And activity number 1 is one that you are going to do on your own. So there is a link in Canvas that will take you to a Snellen chart or an eye chart. Be sure and use the one that that there's a link to, and there's reasons for that. So you're going to do this on your own. You're either going to pull it up on your screen or you're going to print out a copy. If you're on a small screen, you can hold the screen at arm's length. If you're on a bigger screen, you need to step a few feet back. If you print it out, the one that we're linking to is designed to be read from 10 feet away. If you can't measure 10 feet, the average bedroom is somewhere between 8 and 12 feet. So just put it up on one wall, stand at the other wall. It's close enough. So these Snellen charts, I'm going to go back to my whiteboard, are designed to measure visual acuity. And in the US, it's at 20 feet. So you hear the phrase 20/20 vision. The first number says your vision at 20 feet. The second number is typical vision. So 20/20 means that when you're 20 feet away from a Snellen chart, your vision is typical. It's what the average person would see. If it's 20/40, that means that you're standing 20 feet away to see what the average person or the typical person sees at 40 feet. In Europe, these measure the visual acuity is measured based at 6 meters. So they might have 6/6 vision, which means the same thing as 20/20. The way we're doing this is not an accurate way to measure your vision. So if you come up with a weird number, don't worry about it. Exactly how far you stand will affect your results. For example, if you're looking on a screen, the quality of your screen, the distance, that's going to have an effect. Okay, so that's one that you're going to be doing on your own. The next one, number 2, there we go, is looking at pupillary response. And this is going to need a second person and a flashlight. So what you're going to be doing is, I don't care if the other person does it to you or you do it to them, but one person will shine a flashlight in one eye. You'll have your hand or a piece of paper or something up between your eyes. Somebody will flash a light in your right eye, and they'll look at the pupils in both of your eyes. So normal pupils should be the same size in each eye, even if the light is only hitting one of the eyes. Both of your eyes should respond. This is just typical dilated eyes. The pupil's bigger, same concept here. There can be a situation where one pupil is dilated and the other is contracted. If you see this bottom example, I recommend going to see an eye doctor. There are lots of things that can cause this, including, and most likely, not quite doing the experiment correctly. That can certainly cause this, but it could also be something more serious. So that's one that if you do get the weird result, you want to follow up on, but don't panic about it. Okay. Now we're going to do some visual illusions. So there is a approximately 1-minute video for you guys to watch. And what you're going to do is you're going to keep your eyes focused on that square in the center. And then you're going to come back and tell me what you saw. Okay, I've got the first response coming in.
Yeah, that is crazy.
Yeah, it is. It's really bizarre. You can also get similar results by staring at yourself in the mirror, trying not to blink, and your face will, start looking really strange. Yeah, they quit looking like people after a while.
I got the big eyes effect, and then sometimes the mouths would disappear.
Mm-hmm. Yep.
You got these like really long, like noses, like really skinny noses. It's really weird.
Yeah, everybody sees things slightly differently. But yeah, it's always weird. Good. All right. It'd be much more fun if we were in a room doing this at the same time. Lab activity number 3 is visual illusions. So for this one, again, you're going to go to a link and you're going to look at some of these visual illusions. And when you're writing up what you think is going on, you need to think, is the illusion tricking your retina, the eye, or your brain? So you can figure this out by trying one eye at a time. If the illusion still works with each eye, it's working on the brain, not the retina. Or at least that's what it says on the website. I'm not sure about that because I look at this illusion with one eye, and sometimes I see the illusion and sometimes I don't. And it doesn't seem to matter if I've been looking at it or not. So that's another one you're going to do on your own. You're doing a lot of these on your own this week. What's going on here?
It looks like the photo is like being taken like really close to where the bird is. So it's making this like the bird like really big.
Yep. This is something called forced perspective. And a lot of, photographers love to do this kind of stuff. And you'll see some funny things on the internet, but this is also used whenever people find a snake. Snake photos are almost always forced perspective. There was a photo going around recently. Somebody found a really big California king snake. And I could look at the picture and I could tell the guy was holding it almost at arm's length toward the camera. Look how big my fist looks compared to my head. If I'm holding a snake like that versus like this, you're going to think it's a lot bigger if I'm holding it out here. So that's one of my favorite types of illusions.
Yeah, I was going to chime in on this, and I was like, I'm pretty sure that's just a wider depth of field, because otherwise, if you had a shallow depth of field, then all of the background would be creamy and like painting looking.
Yep. Yep, you're exactly right. It has to do with the depth of field. It's going to get more common as digital technology improves, because I've seen some cameras coming along, or I've heard about some cameras coming along that you can tweak the focus even after you've taken the picture, which is just amazing for me. I seem like my pictures are always just a touch out of focus. But anyway.
Yeah, the previous cameras, last generation's cameras were the battle of the foca, the fake bokeh. So you'll see like bokeh is a Japanese word, which is the quality of your out of focus. And with Apple and Google Pixel phones, they have an algorithmic bokeh effect, which really people who are in that tech industry, they call it bokeh because it's fake bokeh.
Okay.
So they purposely, like, they take this image right here that we're looking at and they'll blur the background, but it's not a true shallow depth of field. It's the computer doing it. So you may get like weird artifacting on the sides, like with the bird's hair. You'll get like really weird artifacting.
I'm sure it's going to be getting better and better, though. That's interesting. I didn't know that. All right. The hearing lab, you're going to have to do on your own. Ideally, you'll do it with headphones on, but if you don't have headphones, you can try it anyway. The sounds that you're going to hear doing the hearing test are weird. So just be prepared for that one. You're also going to do the thermo sensation one at home. And when you talk about that one, I want you to use the terms tonic and phasic. So you're going to be immersing your hand in cold or warm water and doing some swapping around and stuff, just to kind of get an idea of the differences there. But we're going to do this one. This is a proprioception test. So what's going to happen is you're going to watch this video. It's less than a minute long. It gives you instructions for how to do the proprioception test. Then you're going to do the test on yourself. Make sure you watch the time, because if you do it for less than 40 seconds, it doesn't usually work. And then we're going to come back and discuss. So I'm going to give you about 5 minutes to go watch the video, do the experiment, and then we'll talk about it. Okay, I see one comment. Did everybody get to get the experience there?
I think, I think my test was biased
Why?
because I worked out yesterday.
Oh,
Specifically, I did overhead presses, so it's my body's going to one, react differently, and two, I'm gonna feel it and respond to it very differently, where it's like, yeah, okay, but the swole effect and the burn, I'm already there, so I don't think it's gonna affect me right, or at
Okay.
least this experiment.
The nice thing about these experiments, in fact, all the experiments we do this semester, there's not a right or a wrong. I'll tell you this right now, nobody fails lab because their experiments turned out weird or didn't work as expected. So yeah, I can understand why a hard upper body workout would mess that one up. Out of body experience. Yep. Yeah, for me, it just feels like my arm is floating.
Yeah, like I'm just watching it do it. Wow.
Yeah. Yep. So this experiment really fooled your proprioception. We also, when we drink alcohol, our proprioception gets all screwed up. And so if you've ever, hopefully watched on TV, but when cops do drug testing, one of the things they do is they have you close your eyes and touch your finger to your nose. You can do it sober. You can't do it when you're drunk because you lose knowing where your nose is. All right. So the last part of the lab is looking at muscle reflexes. Now, I have these links up in Canvas for you guys, but I want to talk about this last one and explain a little bit about what's going on with that one. Most of these are straightforward. You've probably, seen images of the patellar reflex where somebody hits the right below your kneecap with a hammer and the person kicks. You also have a similar reflex in the biceps and the Achilles tendon, which is right above your heel in the very back of your leg. But the last one is the one I want to talk about. Okay. You guys learn what plantar means yet?
Opposite to dorsal flexion, plantar flexion.
Close. It just refers to the bottom of the foot.
Okay.
Is it like when you're turning your foot like in or out kind of?
That's related to that, but plantar specifically means the bottom of your foot. So this reflex, the plantar reflex, the bottom of the foot is stimulated. And in response, your toes are going to curl. It's not an easy one to do, which is why we're watching a video and not trying it. In adults, usually the toes curl down. But That's just usually. The other thing that can happen is what is called Babinski's sign. And in Babinski's sign, the toes curl up and spread out. So if you think about spreading your fingers wide, your toes do that, but they also curl up. And Babinski's sign happens in around 75% of infants. They happen in adults after long periods of walking. Sometimes if somebody does this to you when you're asleep or you've just woken up, or it can be a problem with the spinal cord. So it's part of a series of neurological reflex tests that are done if the doctor thinks that there might be something wrong with your spinal cord. And we're going to watch some of this. That's not what I wanted to do. Next, I want to zoom in. Why does it not want me to zoom in? Okay, well, I'm sorry, it doesn't want to let me zoom in on this page for some reason. Okay. So you've got to be relaxed for all of these. You see how the toes curled under there? And the foot also flexed up a little bit. That is the classic adult response. Oops, that's not what I wanted to do. This is going to be the Babinski sign or this Babinski reflex. And it's really short, but you see how those toes stretch apart? That's that Babinski sign. Okay. All right. You guys have been much quieter than my Monday lab. Oh, is somebody having a birthday today?
Yes, that would be me.
Happy birthday, Brenna. Do you want us to sing to you?
You don't have to, but if you want to, go ahead.
Well, I don't have a great singing voice, so.
You know, I don't either, so.
I've been staring at your tiara, but I couldn't read it for like during last class and this class, and I thought it was your birthday, and I was like, what if I'm so wrong and it's like something else? Happy birthday.
Thank you. My mom told me, she's like, if you're gonna go to class on your birthday, you have to wear a crown, and I'm putting decorations up. And I was like, oh, okay, Mom, like, you don't have to be that extra, but okay, go ahead.
So cute though.
Okay, so do you guys have any questions about what we talked about?
I just have one question about when we meet in person. What part of campus do we meet in? Like, what was the building? Is it the same as lecture or?
Hang on a second. It's all right. Why can I not find mine? Well, I can't seem to find my syllabus for some reason. Oh, because I don't have a syllabus for this class. Right. It should be on Canvas. If it's not, I will put it on Canvas. It's going to be in the science building, the biology building specifically. With that building, there is a big staircase from the parking lot going up. And you are going to go not into the room immediately in front of the staircase, but it's the room to the left of the room immediately in front of the staircase. Yeah, I can see you're trying to figure this one out. So go up the staircase, go left, and it'll say either anatomy lab or physiology lab on the door.
It's in the building where like all of those other labs usually are, right? Okay,
Yep.
perfect.
Yep.
Like in the parking lot, it's like that, if you're going in the parking lot across from Starbucks, like that first building on
Yeah.
the left, like in the corner.
Yes.
Okay,
If you've taken Bio 1 labs on campus, we're in the room right next to that one.
would it be anywhere? I don't know if you'd know, relative to like a chemistry lab?
I don't know where the chemistry labs are.
Okay. I have also a question too.
Yeah.
So since we're gonna be in person and we're starting our lab notebooks, do we need to bring like a new notebook, or if we have a notebook we've been taking notes in through Zoom, would it be that one, or will this be like set up separately? Like, should we have a separate one for our lab notebook?
So here's the thing. I'm going to be taking up the lab notebooks at least twice during the semester. So if you don't worry about keeping track of your notes, if you don't worry about having them constantly with you, or if you're not going to need them while it takes me a week to grade everybody's, then it's fine with me if you do the same notebook.
Okay, thank you.
You're welcome. Anything else? In that case,